All right, this is for the Biotech semester 1 review. Here we go. So, I'm recording it. So first up, as we talk about, let me get this down.
I just screenshotted all those answers.
Good, that's nice. Okay, so it says match the colony to the correct shape. The correct shape is actually on the bottom here as you go across. As it got to places like this, I actually put it on the top. And then if it's margin edge that we're looking at right there, that's going to be in pink. So this is an entire lobated. As you look at lobated. The other one is serrated that I didn't put up there. I'm going to show this down here. It looks like a saw blade. It's a lot smaller, where lobated is lobes. You actually get lobes. Okay. So if we were to do this as serrated, it would be more like this. It looks like a saw blade. Okay. Filamentous is looks like a whole bunch of filaments. Rhinosode, and I hate saying these out loud. This is more like a tree root where it comes down and keeps splitting out, where this one just can be laid upon each other. Raised is just slightly convex. You get an actual, like a half a circle going. These are still both considered entire because it doesn't have any difference in the outside. Undulated, it just is a wavy outside. It's not really serrated yet. Umbinate, got an out. Caravate is an in, so like outie in any belly button. Different ways to look at it from the side. This is a version from the side, so you're looking at the colony from the side. Curled, if you've ever seen moss mold on a tree, that's sort of what curled looks like. It has edges. Spindle, you get this going on. They call it also lens. Irregular, this one could also be serrated. Irregular. Punctiform is a way to say it's smaller than normal. So punctiform is actually smaller than small, and then it goes moderate and large as far as the colony size. And the other one that I didn't have on there was raised margin, where the outsides are raised and the inside are lower. It's not crater form because crater form just has a small crater. Raised margin is just the way the outside is raised, the inside is flat. Okay, any questions with these? Colony and colony names.
What are you talking
about? Goose. Okay.
Wait, I have a question.
Yes, ma'am.
What's the difference between irregular and
serrated?
Lobated? Wait.
Lobated is going to have big lobes. They're coming out. Difference between irregular, this is more serrated. I could have put serrated here. Irregular means I could have some of this going on and then maybe it'd be smooth. So it's not the same all the way around.
Okay.
Okay? So they call it irregular.
Okay.
Okay.
So we have to memorize all these for the test?
Or put them on your 3x5 card.
We get a 3x5 card?
One side only.
No, I would have forgot. Oh my.
Okay, there's some 3x5 cards right there. If you need more,
Just
let me know.
wait, it'll be fine.
Just what they say. I don't know.
I'm putting all these on there. You better believe that.
Okay, then this next part that you need to go over, and there's two parts to it. There's this side and then there's the back side. This is, you need to know how to do gram stain. And you need to sort of be able to explain each part that you're doing. So the first step that really is not on here is you have to heat fix the slide. Remember, you put your sample on and you heated it. Then you put crystal violet over it. And at that time, both of them were purple, whether it was positive or negative. Then you put iodine on it. And at that point, this was purple and this was purple. And then you use alcohol, ethyl alcohol, to rinse it off. The gram-negative became colorless and this one stayed purple. So gram-positive is always going to stay purple, or a violet blue. And then saffron is last step. You add saffron to it. And this one stayed a violet blue, and this one turned red, so you could see it. Because the colorless is hard to see underneath the microscope, but with the red coloring, you could see it under the microscope.
Okay, wait, I'm not going.
Nope, you're fine. Let me know if I'm going too fast.
Why does that big line say purple if it's like colorless and red?
Because this whole thing for gram-positive is that purple or violet blue color. It doesn't change.
It never changes.
Nope.
And then it only changes if it's gram-negative?
Right.
That's how you know if it's one or the other.
Right.
What are we
But
saying?
the final stain for gram negative is so we can see it, because colorless is going to be hard to see under the microscope. Okay, we okay to go forward?
Yeah.
The next page is more of the same, just breaking it down a little bit more so that you can talk about what's going on. It's still the gram stain procedure. So again, the first step is actually fixing to a slide. That's the first step. And you have to heat fix it, remember. Okay. And I got off a little bit here, so just pretend that it's all one up. So, because I put the heat fix to a slide here when it should have been up here. So the first one is the initial stain, which is the crystal violet. And both cell walls affix the dye. They both take in the dye. They both look purple. Then the next, you flood it with iodine for 1 minute and rinse. So now this is where you got the mordant is used to fix the stain. So just remember it's one up for all mine because I got off by one. But the dye complex is trapped in the cell wall and doesn't affect anything here with the effect of iodine. That's because of that, when you get to the next step, which is where you use the ethyl alcohol or ethanol, you put the ethyl alcohol in here, it decolorizes the gram negative. And the gram negative outer wall membrane is weakened because of that, and the cell in the gram positive still remains purple. Then the last step is your saffron, and with the saffron is added, this is the counter stain, and with the counter stain, this one, the red dye is sort of masked by the violet because the violet is darker, so you really can't see it. So it still looks purple, but the red stain here stains the cell so we can see it. Sorry about me getting off one here. All right, on the next one, you're going to draw colony profiles, and you'll be able to name the profiles.
Wait, sorry. Could
Nope, you're
you
good.
scoot it over so we can see it? And yeah.
Is this going to be up on
Yep,
canvas?
yep, I'm going to put it up on canvas, so if you get behind a little bit, you can go there, listen to it again, or mute it and just watch me,
It's like an audio book.
something like that. Just watch it over and over again until you can't stand it anymore. Okay. We okay to go forward?
Yeah.
All right, I forgot to put the raised margin on here again, and it is on the test, so I went back and put it back on. But you need to be able to draw or identify each of these.
My goodness.
Just remember, crater form has just got a little crater in the center, where raised margin has a large area in the center that's
That's
lower.
all it knows.
So what's the difference between the convex and the raised? It's basically the same.
So
as
raised
that.
is just barely raised. Convex, you actually got almost a half a circle. And pyruvate, you've got half of an oval. Okay, and then we get to the next one where we've got to identify a bacteria cell, the parts of it. So the little dots are the ribosome, the nucleoid is in the center. The cell wall is the outer wall. The plasma membrane is the inner wall. And the cytoplasm is the fluid in the cell. And some of them have the flagella, which is how they move about.
You do people.
Miss Howell,
Yes?
I need Elizabeth Fox to come to the attendant's office to check out.
Okay, it'll be up online.
Thank you.
All right, we okay to move forward? All right, so on the next part here, we're actually doing calculations. So you need to be able to do molarity, which is just moles over liters. So if you have moles, great. Here we have milliliters, change it to liters by dividing by 1,000. So it's divided by 1,000 to get it from milliliters to liters. Then take moles divided by liters and you got your molarity. So molarity is equal to moles divided by liters. On the second one, you're given grams and you're given milliliters. So the first step is to add up the mass of calcium chloride from the periodic table and get the mass. Then take the grams, divide it by the mass you added up, and that's equal to 1 mole. Once you've plugged that through your calculator and got this, then again divide this by 1,000 to get it from milliliters to liters. Take moles divided by liters and you've got molarity again. On the next one, we're working at a percent. We're trying to find percent. This is basically all the C1, V1 equals C2, V2. So you're doing the same thing. You're just solving for how much you're going to put in. So for if you need 5 milliliters made of a 40.5 solution, and you start with 100%, you just take the 4.5 times 5 and you divide it by the 100 from the other side to get how much you need to have. You take this ethanol and then you dilute it to the 5 milliliters to get your 4.5%. Okay, any questions with that? Okay, given 98% instead of 100 now, you need to make a 15% solution. You need a volume, say 1 liter. So if I want to make 1 liter of the 15%, again I'm using C1V1 equals C2V2, plugging in that I'm going to have 15 times 1 divided by 98. So in order to make this 15% solution, I'd need to put 0.153 liters, which would be 15.3 milliliters, in a volumetric flask and then dilute it up to 1 liter.
What does the C stand for? Concentration?
Concentration and volume.
So the concentration will always be a percentage?
It can be, or it could be a molarity. So concentration could be a percent, molarity, it could be a molality, it could be anything that we use for concentration. PPT, parts per 100, parts per 1,000, parts per billion, anything like that.
Okay,
Whatever
so
it's
because this gives us 2 concentrations.
2 percentages. We need one more thing, which is we need a volume. So
then
pick
you
something
said-
easy, like a liter.
Okay.
Because then you can say, oh, I make that 15% solution. I make a liter of it by doing this.
Okay.
By measuring out 15.3 of the 98% HCl and diluting it to 100 or 1,000 milliliters. Okay, so same thing with making the agar. We've made agar before. So on the bottle it says to mix 45 grams to make with 150 milliliters of distilled water to make the agars. So we need 275 milliliters of agar, and we don't want to waste anything. So we set up a ratio. 45 is to 150 as whatever is to 275. We solve for x here. You're going to get 82.5 grams of agar that we would dilute to the 275. Okay?
We have to word it like that.
No, you could say, how else would you word it?
So I'm just curious, like, do you have to put all of the numbers in there? Or can you just give the answer?
So if you said just 82.5 agar powder, how much water are you going to use? So usually add that in somehow. You weigh out this and then dilute up until however you want to put it. Okay, the next one, determine the number of moles if you have liters and you have molarity. Well, if you remember, molarity is equal to moles divided by liters. So if we're looking for moles, moles are going to equal molarity times liters, right? So we just take molarity times liters. Liters will cancel, and we've got moles. On the next one here, hydrogen bonds work only between hydrogen, nitrogen, hydrogen, oxygen, or hydrogen and fluorine. It's an interaction between oppositely charged ions, so these would be negative, and this would be positive. Hydrogen attracts to just those three to make hydrogen bonds. Ionic bonds are electrons are transferred from one atom, not one electron, one atom to the next, making one positive and one negative, and that's why they call they're electrostatically attached. Covalent electrons are shared. And then this bottom part here, proteins are made up of amino acids held together by peptide bonds. Lipids are used to store energy, and a major part of cell membrane is mostly carbon and hydrogen. Carbohydrates are usually carbon, hydrogen, and oxygen, and they're used for quick energy. Nucleotides are 5-carbon sugar pentose rings with phosphate groups and a nitrogen base. Are we okay? Nope, still writing.
Man, this class is so quiet.
We okay to go forward?
Yeah.
Next part here, what type of macromolecule is made up of amino acids? That's proteins. Amino acids are held together in between. They're used to store major energy. That's lipids. That's also known as fats. That's lipids. Describe a solution. It's a solute and a solvent. A solute is dissolved in a solvent. Usually a solid and a liquid, but it can be a liquid in liquid or a gas in a liquid. What happens to a solution when the solute increases? It becomes more concentrated. When it decreases, it becomes less concentrated. Okay, now this is important when you come to this one. You need to look at the units. So milligram and milliliter are off by 1,000 each, right? So 1,000th of a gram and 1,000th of a liter. The gram and the milliliter, this one is a small unit, this one is a bigger unit. This is a gram and a liter. Micro means 10 to the negative 6 to a liter. Micro to micro, that's 10 to 10, or 10 to the negative 6, 10 to the negative 6. So it's basically like saying gram per liter. So that would be equal to 4.3 milligrams per milliliter, or equal to 4.3 grams per liter. Okay, so we could fix it that way. And then kilograms per liter. So this one actually is the most concentrated because it's the most in a liter. This is the least concentrated. Micro is a lot smaller than liter. So it's the most dilute. Okay. As long as both of them are increased or decreased the same, then the number, if it stays the same, they're equal to each other, as in concentration. Okay, on this next table is just filling it out from the chart. So sodium is number 11. That's its atomic number. It's also the number of electrons. As long as it doesn't have a charge here, the number of protons is the number of electrons. The mass number is the number of protons plus the number of neutrons, which is 23 here. Or we can go the number of mass number minus the atomic number is the number of neutrons. To show nuclear notation, we put the mass number over the atomic number and put the symbol. Yes?
I think your last one is wrong.
Which one?
Silver.
It is supposed to be silver, yes. It is wrong. I did a copy. So this should be silver. and this should be Ag. Thank you. Okay, so if the protons and the electrons are different, then remember electrons are negative, protons are positive, so whatever you have more of tells you what charge you have. All right. Anybody have a question with this part? All right, the next one down here says, what information do you need to help determine the number of moles in a solution? You need the concentration in molarity, and you need the volume that you're using. And then you would take molarity times volume to get number of moles. Okay, some characteristics of bases and acids. We did that a while ago. A base, just remember, blue litmus paper, bitter, and it's OH. An acid is red, sour, and contains hydrogen. And we go back to aseptic technique, things that you need to do to prevent contamination while you're working with bacteria and bacterial agar. Remember, we needed to flame the inoculation lube, not set it on the counter. Flame the mouth or lip of the container that we're going to use so there's no airborne contamination. And then wash your hands, wear gloves, wipe down the counter with 10% bleach or 10% ethanol. Yes?
Is this doing canvas? This
No.
review?
Just to help you with the test. Okay, we okay to go forward? Maybe we need a few more minutes here. Okay.
What? How's it going?
I'm not even writing anything down. Okay, we good to go? Or we need a few more minutes? Okay, we're still working. Okay, Caleb, move forward. Okay. Next part here. If you want to make 60 milliliters of a 30% stock solution, from a stock solution, how much stock would you need? So you've got to know that the stock solution is probably 100%, hopefully, we're assuming here. And we're still using the C1V1 equals C2V2. So plugging in, we're solving for X. We'll divide both sides by 100. We'll need 18 milliliters of stock diluted to 60 milliliters to make that solution. And then how much water would be needed to make 100 milliliters of 40% stock? Well, we could go about the same thing again, but we're going to end up needing 40 milliliters of the stock solution. Therefore, we need to dilute up to 100 by adding 60 milliliters of the distilled water. Okay, on the next, you're supposed to identify the organelles and the correct description of them. The one on the left is an animal cell, the one on the right is a plant cell. So there's a couple of things I added in here. This is a cell membrane, this is a cell wall. This one from our other is the cell membrane. Nucleus is here. It didn't have you do the nucleus, or it did, but it didn't have it pointed to the right thing, so I put it there. Over here, A is cytoplast, chloroplast, endoplasmic reticulum, then ribosome, Golgi apparatus, I'm not going to say that right, large central vacuole, and then the mitochondria. Some other things that we need to know with this are that the endoplasmic reticulum makes proteins or lipids, and the Golgi apparatus packages proteins and lipids for their transport in or out of the cell. And then the nucleus holds a majority of the DNA. So those are some things we need to know about those cells.
I have a question.
Yes.
What's the difference between the mitochondria and the chloroplast, like the way they look with it?
So the chloroplasts actually will have, they don't have this squiggly nuclei like thing in the center. They actually have like the squares here. So they look square. And in an animal cell, you won't see the chloroplasts. You see they're only over here.
Okay.
Same thing with the F, which is the large central vacuole, is only in the plant cell.
But all cells have
But all cells have
a
everything else.
mitochondria.
Yep. Okay. Then what does the mitochondria do? It supplies energy to the cell, so all cells need energy in order to function. So that's why they all have one. And how is the cell shown different from prokaryotic cell? It has a nucleus. Okay. And then the organelle responsible for converting sunlight into sugar is the chloroplast. That's how we can get sugar cane and food because of the plant cells. And the last thing down here is just converting or knowing what the base units and the units are for the metric system. What do they stand for?
Sorry, I have another question.
Yes, ma'am.
Like when you're looking at it,
Mhm.
the nucleus and the large central vacuole, like, so it labeled it wrong, is that what you said? So like the nucleus is only in the animal cell, but then the plant cell.
No, there should be a nucleus over here too. See it?
The big circle dot thing.
Yeah.
Okay. So that's the difference between the vacuole and the nucleus, as you'll see.
This one has a vacuole, this one does not. This one has a chloroplast, this one does not.
So is prokaryotic a plant cell?
Prokaryotic is a plant cell, but it doesn't have a nucleus. Okay?
Okay, thank you.
We okay here? This is just metric. Seen it forever, I think, I hope. Okay, going on the back side of this. This is back to lab now. A meniscus is considered when making what kind of measurement? Volume. Okay, reading this, you should read right here. It's right on the line, so it's 24.0, and it didn't give me a unit here, but I'm guessing it's milliliters. Okay, then you should be able to look at the pipette, because we've been reading pipettes a lot now, and be able to tell me, if I give you the P whatever on the top, what this represents. So if there was a 1 here, that would be 1,000, so this is the next step down, so it's 150. This is between, exactly between, so it's 0.5, or 155. This one right here, the first one is occupied. It's a 200, so it can be 1 or 2 or 0. So I've got 155.5. And then the last one is a P20, so this is 15.5 and then 5. And then we've got the ones on the bottom here, where we're actually giving a picture of what's going on there. Have to sort of look really carefully. But this is 6.8, because this is between 8 and 9, and then 4. And then this one is 132.4. And then this one is 260, and the thing's right here. 2 or 4, I would take either one of there, the 2 or the 4, because I can't see how it's divided up, if it's worth 2 each one or just 1. Okay. Then you should be able to look at some pipettes and be given an amount and say, which one do I need to go and grab? So should I grab a 20, a 100, or a 200? Well, if I'm doing 75 microliters, I probably should grab a 100, because that's within. I could use a 200, but more accurate would be the 100. Then the basic safety rules for lab. gloves, goggles, aprons or lab coats, nor horseplay. Clean up with 10% bleach or ethanol.
Move.
Okay. Metric standards, masses, grams, volumes, liters, time in seconds, length in meters. And then we're reading again what this should represent. So again, this is the thousandths place, the hundredths, and the tens. So that's what I get. This is the ones, tenths, 100ths. And then this one is the 10, ones, and tenths. Okay. Oh, I didn't do this one. So 0.435 kilometers to decimeters. I always go to the base unit first. So 1 kilometer is 1,000 meters. 1 meter is 10 decimeters. And then you would just figure it out, which would be 4.35 times 10 to the third decimeters.
It's 4,032.
Yep. Okay, next one. How does flaming the mouth of a test tube flask help prevent contamination? It prevents airborne contamination. So any bacteria that's floating around in the air, it's got on the bottle. By flaming it, I've killed it. It doesn't contaminate my sample. How does a single colony form? Physical separation allows for a single bacteria to form a colony by replication. What personal protective devices are required in our labs? Gloves, goggles, aprons, or lab coats. How many millimeters are in a centimeter? 4.5 centimeters, again, 4.5 to 1. And then we're looking for millimeters, so in 1 meter there's 1,000 millimeters. Just basic conversions. Okay, any questions so far?
So easy.
Make sure you stop me if I start going over you. Okay, last page. On the last page, at the top here. What does molarity mean? It means concentration in moles of solid over liters of solution. And then putting 4.5 moles of glucose in 1.5 liters. What's the molarity? Just take moles divided by liters and you've got the answer. At a given volume, what happens to the number of molecules of a solute as the solid concentration decreases? The number of molecules decreases. As the solid concentration increases, the number of molecules increases. And then what shape do you make when your inoculation lube when you're streaking a plate? A Z shape. And when to use a aseptic technique when you're working with a live culture to avoid contamination. Now, the thing I forgot to put on there was the bacterial shape morphology. So I put that in, and you can sort of draw them in or do whatever you need to, because you'll need to be able to identify each of these and name them. So you've got the spirula. Sometimes they have, my brain just left. Sometimes they have flagella, and sometimes they just look like a zigzag kind of thing going on. The spiral shet actually looks like a coil. Okay, so that's the difference. This is more of a zigzag. This is a coil. Coccus is just a circle. Coccus bacilla is between the coccus and the bacilla, which is more like a pill shape. So you end up more with an oval. And the
Can you scroll
fusiform
up a little bit, please?
could what? Up or
Scroll
down?
up a little bit. Or down a little bit. Perfect, thank you.
Uh-huh. The fusiform bacillia is just like you take and you stretched it out. The vibro, I think of like a telephone. It vibrates here, and it's got a little flagella. We okay to move up, or you need a little bit more time? Now move up to here. Moving up to there. Last part down here at the bottom. You got streptococca, that is cocci in a line. Diplococci is when you have two. This is pneumonia. So if you've ever had pneumonia, that's Diplococcus. Staphylococcus looks like grapes. They sort of go in a grape bunch. Filamentous is just filaments over at the top of each other. And then Diplobacillus is just two pill shape hooked together. And I didn't do a very good job at pill shape, but basically pill shape. Two of them hooked together. Okay. And Streptobacillus would be all of them in a big line. Staphylococcus would be making a grape shape with the bacillus shape. Okay. All right, and that's it. So that gives you almost 30 minutes to work on whatever you need to.
Oh, yeah.
Okay. And then just study hard for the test. Put on your 3x5 card, whatever you need for the test.
We didn't have any sub-assignments, right? So there are any on here. We don't do them. Like, you don't.
Did we just get 100% on this?
I'm in the wrong place. Let me look real quick.
Okay. You draw that. There's like a bunch of them in here.
Well, you should have done the human experimentation nearpod with bioethics.
Oh, yeah. Those are quick and early.
You should-
Is that one on this semester or is that one last semester?
That's this semester. Oh, that's last semester. Sorry. So this semester, grab your biotechnology books, November 21st. I don't think I had you do that.
Because I remember the days that you worked on, we just did like work days.
Yeah.
But I'm just curious.
So let's put that out at 0. See what else.
No, one more. So I have it from this like job. I have it all the way back to it, but I don't remember.
Three pictures. All of us.
Would you mind going all the way to the top of that page?
I'll see what you say. I have all these notes.
Thank you.
Mhm.
Really? Yeah,
Yeah, I don't
I'm
think
sorry. You said
I don't
that
think
earlier,
we
like,
had any of
goose.
those. Let me
Goose?
take them
Sounds
off.
weird.
Okay.
Says goose.
I'll just unpublish them. As long as nobody's submitted, we can go.
Goose, goose. Goose.
We did do one rate the sub.
Yeah.
Okay, so those should be off of yours now.
So we did the exploring how buffers work. Like, did we do
Yes, we did that one.
Really?
Was there a paper for it?
I don't have it in mine either.
And it says it's locked too.
On which one? Exploring how buffers work?
Yeah, it says it's locked November 30th.
Could you also look at the submission for journal entries too?
Where are you looking for that one?
Just on my grades, like the thing like that. This is a paper.
Yeah, mine says it's missing too.
What's it called again? I've got demonstration
But
of,
it's like a whole lab.
Yeah, it's exploring how buffers work.
we did buffer
I thought we did
efficacy.
one.
I know we did a buffer
Yeah,
one,
we
but wasn't
did.
that in the
The buffer
lab?
efficacy, yeah. That's
But
why
this
I'm
is
trying
like
to
a
find
whole
the other one.
separate lab.
Let me find it. Don't submit anything, guys. Don't submit. Because if you submit, I can't get rid of it. I can't see it. Let me see if I can go to your grades and see it that way.
I don't know. I just kind of worked on them like as I went. It didn't take too long. I just worked on them. Like there's only 20. So it's not that bad. I mean, it's so tedious.
I'm not seeing that, even on your grades here. Do it one more time. That's what it's called. It's so weird. I've been having problems with Canvas all day long. Going back through it again. Exploring how buffers work.
It says it was due November 25th,
Interesting.
and now it's locked. Do we get the front and back of the index card or just one?
Just one side.
Just one side?
Yep. Be glad you get one side.
Is it like
A whole
You want the
packet
whole
just
science?
as answers. Pass this.
question is, what am I willing to memorize and what am I not willing to memorize? What if all the quiz?
There it is, right there. I found it.
Yeah, I think you like that.
Okay, I unpublished it and set it on a 0, so if it comes back either way, it's
Look at
gonna
this.
affect you. Okay, let me know if there's anything else like that, because that shouldn't have been there.
And then just ignore all of the.
They should be gone. I unpublished them. So let me know if there's anything else weird like that.
Yeah.